Hello, my name is Ray Tao Lo, and today we are going to talk about the functions of different genes associated to life. And is it possible to modify humans and have an eternal life? If you don't know me, I'm the director of Biochemistry and ambassador of Berkshire in the Youth STEAM Advisor. In modern days, gene modification is the hottest topic in biochemistry, and so we are going to discover why is genes important and how does it affect our lives. Just to specify, the purpose of today's video is to awaken your curiosity towards science. So if you don't understand something or want to investigate more, which I recommend you to do is to search it up on Google for further informations. So let's get started. So what is genes? Genes are small units of heritage found in our DNA. They contain instructions for how our body grows, develops, and functions. So it's basically a blueprint of our body. Do you know that 99.9% of human genes are the same as others, but tiny difference makes us all unique? DNA is made up of only 4 bases: adenine, thymine, cytosine, and guanine. And these 4 bases can pair off with sugar and phosphate to form genes. These are in complementary pairs A and T, C and G in the DNA, and these two pairs arranged in different sequence, and is the reason why we are the person we are now. So humans have about 20,000 genes used to produce protein, and each gene carries out different functions. For example, there are genes that are crucial for basic survival, like TP53, which provide instructions for making a protein called P53, and it can suppress tumor and regulate cell division from dividing too fast. It can also determine whether the DNA will be repaired or let this damaged cell undergo self-destruction. There are genes that are related to immunity, such as HLAs, which helps the immune system distinguish the body's own protein from proteins made from foreign invaders like virus and bacteria. Hence, we can eliminate them before damaging the cells as early as possible. Lastly, the genes that are related to intelligence and brain functions, like SHANK3, that provides instruction to make a protein that is found in many tissues but most abundant in brain. Scientists find out that they play a role in the functioning of synapses, which are the connections between nerve cells for electrical signals to transfer. What's interesting about genetics is the mutation, which has two sides of consequences, which could either create a detrimental effect or make a person superior, like the EPAS1 gene, which is often called the super-athletic gene, which are mostly found in the shepherd people who live in the mountains. The gene can ensure efficient oxygen transport without the thickening of the blood and help them adapt to high altitude and higher endurance. Myostatin mutation is a rare condition that can reduce body fat and increase muscle size by approximately 2 times, like a superhuman with extraordinary strength. Only 0.0004% in the population have this condition. For example, Liam Hoekstra had the six-pack and could lift 5-pound dumbbell at just 3 years old. On the other hand, SIRT6 can regulate the aging and metabolism process, and APOE can decrease risk of Alzheimer's disease and protect from decline in episodic memory function, which is basically the ability to recall memories. For example, Jenny Coleman is the longest living person on Earth, which is 122 years old. Researchers think that she has a mutation in the genes of FOXO3 and other genes, which contributes to her stress resistance and longevity as she remains good condition and physical health past 100, which is an incredible achievement. The LRP5 gene can cause an uncommon disorder in which the bone density is greatly increased, making the bone very strong and resistant to fracture. It is also a very rare case, and less than 0.0001% of people got it. The real example is in 1994, a person is involved in a car accident, which is serious enough to cause multiple fractures. However, he walked out unscratched. The man was later referred to a researcher and found out his bones was 8 times denser than a normal person's bone. Isn't that a ability that we all want? So I think you have all watched fiction movies which people get genetic mutations, making them have superpowers. The most fascinating ability I've seen is to live eternally, as that means you have infinite time to do whatever you want. So here's a question. Is it possible to enhance a person by genetic modification or other methods and potentially live forever? I can't give you a definite answer, but the answer is probably yes, as scientists are already on the investigation and have some progress on it, which could either make you live eternally or slow down your aging process. The picture I've shown you has some hallmarks on it, and scientists have figured out that these have contribution to our aging. And here's the methods that can either slow down our aging or eventually solve the problem. Telomere length and telomerase activation. Telomere are protective caps at the end of chromosomes that shortens as cell divides. When they become too short, cells can no longer divide, leading to aging and cell death. But telomerase is an enzyme that maintains telomere length. So scientists have a brilliant solution. They find out telomerase gene therapy or specific food and vitamin D are proved to increase activity of telomerase and lengthen telomere, so they can potentially slow down our aging process or even anti-aging. Calorie restriction is a dietary regimen that reduces calorie intake by 20 to 40% without causing malnutrition. This practice improves metabolic health, reduces oxidative stress, and lowers inflammation. If you're unfamiliar, oxidative stress refers to an imbalance between free radical and antioxidant in the body. Additionally, calorie restriction is believed to slow aging by boosting sirtuin activity through the activation of AMPK and increasing NAD plus levels. NAD plus levels supports longevity by enhancing cellular energy production, facilitating DNA repair, and activating sirtuins, which play key roles in stress resistance, mitochondrial health, and regulating aging process. Sirtuins contribute to cellular repair, metabolic regulation, and epigenetic control, further promoting health and longevity. Senescent cells and aging. Senescent cells are those which have stopped dividing but do not undergo programmed cell death, also called apoptosis. Over time, these cells accumulate in tissues and release inflammatory factors, contributing to aging and the onset of age-related disease. Senolytics such as dasatinib and quercetin are compounds that selectively target and eliminate senescent cells, potentially improving tissue functions and extending lifespan. For example, the BCL2 protein family, which plays a role in regulating apoptosis, is often overexpressed in senescent cells. Senolytic drugs like navitoclax inhibit these proteins, triggering the self-destruction of senescent cells while leaving healthy cells unaffected, as they do not rely on these pathways. So, epigenetic modifications and aging. Epigenetic alterations are changes in the chemical structure of DNA that does not affect the DNA coding sequence. These changes occur when chemical groups, such as methyl groups, are added or removed from the DNA, or when histone proteins that bind to DNA are modified. These modifications can result from environmental factors, including diet, exercise, drugs, and chemicals, and may contribute to aging and disease risk. They can also be inherited from one generation to the next. Yamanaka factors OCT304, SOX2, KLF4, and CMYC are a set of four transcription factors that can reprogram somatic cells into induced pluripotent stem cells. iPSCs are pluripotent cells capable of differentiating into any cell types. By resetting the epigenetic marks in cells, Yamanaka factors can reverse cellular aging and rejuvenate cells without altering the DNA sequence itself. In mice, partial reprogramming has shown promise in rejuvenating cells without causing cancer. CRISPR-based epigenome editing allows precise modification of methylation and histone marks at specific genes. By altering these epigenetic marks, which influence gene expression, CRISPR can change how DNA is read without modifying the underlying genetic code. This technique has the potential of impacting aging and disease treatment by controlling which genes are activated or silenced. So, hooray! We have finished this lesson. Hope you have learned a few more things than the person you were a few minutes ago. Hope you enjoyed this lesson and don't forget to subscribe to our channel. Also keep an eye on our website and our events, including the Bio Bootcamp, which we will be teaching you a lot of things in April and May. And so, bye bye.